So today we'll start with the topic of vaccines, and vaccines are very important in fighting diseases. They are actually the prophylactic measures. That means a measure that we take before the disease starts. And early roots of vaccination are embedded in the trials of Edward Jenner and Louis Pasteur. Edward Jenner in 1796 actually observed that cow mates contracted a mild disease called as cowpox, which lasted for 10 to 12 days. However, this cowpox disease prevented smallpox disease in these cow mates. They never got the smallpox disease. Jenner observed this and he opined or he thought that because of this disease, the mild disease cowpox, the cow mates they are protected from the disease smallpox. However, in order to check his theory, he injected cowpox pustule in a boy. The boy developed cowpox, and after 10 to 12 days, he recovered from the disease. He again injected the same boy with smallpox pustule, and the boy did not get any kind of smallpox disease. There were no symptoms, and he was healthy. Thus, he proved his theory that cowpox somehow protected the boy against smallpox. Nearly 90 years later, in 1870s, it was Louis Pasteur who was working with chicken cholera. He saw that the old cultures of cholera bacterium, when they are injected into the chicken, they develop a mild form of disease and do not die. When these chicken were again injected with the virulent form of the bacteria, the chicken did not get any disease and were protected from subsequent challenges by the bacteria, by the cholera bacteria. He actually formed the basis of live attenuated vaccines. According to him, the age of the cultures attenuated them. That means that cultures had lost their activity or ability to cause disease, but they retained their capacity to uh, generate immunity. That is what attenuation is. And many of the vaccines that we use till date are based on this principle, the attenuated vaccines. Pasteur continued his work with Bacillus anthracis, the anthrax bacterium, and also with the rabies. So he actually attenuated the anthracis strain and proved that attenuation could be achieved. Right now, there are lots of vaccines that are being used for different diseases like diphtheria, measles, mumps, polio, tetanus. In fact, smallpox vaccination is no more used because of vaccination efforts, the world was cleared of smallpox disease in 1970s, and from that time onwards there was no smallpox. And because of the vaccination efforts, the world is soon going to be free of polio, because in 2018 and 19 there were as low as 30 cases of polio throughout the world, and within next few years it is expected that the polio will be eradicated from the world. Costly, human trials are required, and there could be a vaccine that could be developed, but there could be adverse effects, so it cannot be used. Before going to the actual development of vaccines, we need to know about the concept of passive immunization and active immunization. Passive immunization is transfer of preformed antibodies. That means antibodies are generated in another organism and then they are injected into the host, or the recipient does not produce those antibodies, but they are transferred from some other organism. Like you have antivenoms, they are developed in horse. The horse antisera is used as antivenoms. It does that means that if somebody is bitten by a snake or some kind of a venomous animal, this antisera is given, but it does not induce a long-term immunity. Whereas active immunization is long-term protection, the development of immunologic memory, and there is actual exposure of the pathogen to the host, so that a long-lasting immunity is generated. And vaccines are based on the principle of active immunity, not on the basis of passive immunity. This is a list of the passive immunization antivenoms or that are available, like horse antivenin, horse antitoxin, so on and so forth. Whereas this is the recommended childhood immunization schedule. Most of the countries of the world follow this schedule, like in India also. Apart from hepatitis B at the birth, we also give BCG, rotavirus, diphtheria, tetanus, DPT is also given. Haemophilus influenza is also, pneumococcal has also now been introduced into the immunization regime in India as well. We have inactivated poliovirus, which is IPV, injectable poliovirus or polio vaccine. There is also oral polio vaccine, which is live attenuated. We'll discuss what are live attenuated and what are the killed vaccines. Influenza vaccination is not included in India, whereas MMR, mumps, measles, and rubella is present. Varicella zoster is for chickenpox, that is also available, and hepatitis A is also given. There are chances of adverse effects of vaccine in small amount of population. This is the case with any treatment or drug. However, if the benefits to the population they outweigh the risk of the side effects, vaccines must be used and provides herd immunity. Now, actually, the concept of herd immunity is very important. This means that if in a population of at least, say, let us talk about a population of 100 people, if 96 or 95 or 98 people are immune to a particular disease, that means the chances of them being a reservoir of the disease are very low, and those 4 or 2 or 3 people who are not actually resistant or immune to the disease will not get the disease, because there won't be any source of the disease. The chances of those individuals getting a disease will be 2%, as low as 2%, because 98% of the population is immune, which actually means that in a herd, in a larger population, if a larger percentage of population is immune, the smaller population also remains protected from the particular disease. So there might be some people who cannot be vaccinated, and also because you got the vaccine doesn't mean it worked in you. So there are the designing of vaccine actually involves the development of protective immunity, and it must produce immunologic memory. So these are the vaccines that are the different types of vaccines, like live attenuated, them one by one, and then we'll come to understand what each vaccine does. So as we discussed about the Pasteur's experiment, which created a live attenuated vaccine, microorganisms can be attenuated so that they lose their ability to cause significant disease, but they retain their capacity for growth in the host. So bacteria is grown for a prolonged period in adverse conditions. Those that survive will not be suited to grow in better conditions in the host. A simple example of this type of vaccine is BCG vaccine. BCG is actually an attenuated strain of, not of Mycobacterium tuberculosis, but that of Mycobacterium bovis, that causes TB in bovine animals. So it is called Bacillus calmette-guerin. These were two scientists, Calmette and Guerin, who actually cultured the bacteria for 13 years in increasing concentration of bile salts, and this actually attenuated the bacteria. It could not cause any disease but protected against tuberculosis. So this actually gave rise to one of the live attenuated vaccines, which is BCG. For viruses, viruses might be grow in cell type which is not normal to them. They accumulate mutations that might weaken it, and then it has to be subsequently checked whether it has been attenuated or not. Like measles, mumps, rubella vaccine is an example of some kind of bacteria. So attenuated vaccines, they have the advantages. That means they can grow in the host for a longer period of time, and therefore the exposure of the host system to the antigens of the pathogen is for a longer time, and therefore no booster doses are required. It produces T cell memory as well, and it's good for distribution in third world countries, because no booster doses are required, because it has been recorded that almost 30% of the population in the third world countries don't turn up for the booster doses. They come for a dose and then they don't come for another dose. So such kind of vaccines are very important when only one, because only one dose is required. Whereas if you talk about the disadvantages, it is possible that the live attenuated vaccine, of course, is a live organism. It can revert to virulent form. The chances of getting a virulent form in polio virus, the oral polio virus vaccine, is 1 in 2.4 million. There are complications like some cases in measles vaccines are actually associated with encephalitis, and there are certain dangers from remaining unvaccinated and getting disease is much greater than complication to these proven vaccines, which is of course true, because the complication chances of complication are very low, whereas the chances of getting these diseases is very high. So these are some of the dangers and advantages that are associated with live attenuated vaccines. So I'll talk about the inactivated or killed vaccines next. The inactivation of pathogen by heat or chemical means, generally chemical means are preferred